Oracle, Arizona. September 26, 1991. In the foothills of the Santa Catalina Mountains, north of Tucson, there is a structure that looks like nothing else on Earth. Pyramids of glass and steel, white space frame arches, domes, greenhouses layered and connected, just over 3 acres of enclosed space, about 1.27 hectares. Every surface sealed against the outside world. 8 people walked in through the airlock doors that morning. A crowd outside cheered, cameras rolled, the doors closed behind them. The agreement was this: 2 years completely sealed, no air from outside, no water from outside, no food from outside. Whatever they needed to survive, the world inside would have to provide. The project was called Biosphere 2. The name was intentional. Biosphere 1 is Earth. Biosphere 2 was the attempt to build another one, smaller, controlled, replicable. The stated purpose was to demonstrate that humanity could construct a closed, self-sustaining ecological system, a world that worked by itself, because if we were ever going to put permanent human settlements on Mars, we would need to know how to do exactly that. The structure had taken 7 years to build, and had cost approximately $200 million, funded largely by an oil heir from Texas named Ed Bass. The intellectual architect was a man named John Allen, writer, systems ecologist, founder of the Institute of Ecotechnics, whose vision had been to create a miniature planet and see if it could sustain itself. Inside there were 5 biomes: a tropical rainforest with palm trees growing to 15 meters, a savanna, a fog desert, a mangrove wetland, and an ocean. 850,000 liters of salt water, with a wave machine to replicate tidal action, with living coral reef, with fish. Alongside these, an intensive agricultural zone modeled on Mediterranean farming. Goats, chickens, pygmy pigs, roughly half an acre of intensive crops, calculated to feed 8 people for 2 years. The 8 were not random volunteers. Roy Walford, 67 years old, was one of America's leading gerontologists, a physician and researcher whose work on caloric restriction and longevity was internationally known. Jane Pointer handled agricultural botany. Tabor McCallum was the analytical chemist. Mark Nelson studied ecological systems. Abigail Alling was the marine biologist responsible for the artificial ocean. Mark Van Thilo was the mechanical systems engineer, the person who kept everything running. Sally Silverstone and Linda Lee managed agricultural research in the desert biome, respectively. The system contained approximately 3,800 species of plants, animals, and insects. The food web had been mapped in detail: bees for pollination, earthworms in the soil, 4 species of cockroaches, imported specifically to decompose organic matter. The ecological model had been refined over years. There were also the lungs, Biosphere 2's most unusual engineering feature: 2 large hemispherical chambers, connected to the main structure by underground tunnels, each sealed by a large, flexible rubber membrane. As the air inside expanded and contracted with temperature changes, hot Arizona days, cold nights, the membrane moved in and out, equalizing pressure and preventing the glass panels from being destroyed. The structure breathed. Everything had been modeled. Everything had been calculated. On September 26, 1991, the doors closed, and the experiment began. Month 1 was fine. The crops grew. The coral reef was stable. The rainforest canopy was expanding. The agricultural zone was producing. Month 3, the headaches began. Low-grade, persistent. The team attributed them to adaptation stress. By month 6, the oxygen sensors showed something that couldn't be attributed to stress. The atmospheric oxygen concentration had dropped from 20.9% to approximately 19.4%. The plants were growing. By all accounts, the photosynthesis was happening. The oxygen should have been stable. Roy Walford began documenting the physiological effects with the precision of a research physician. Cognitive slowing, fatigue that sleep didn't resolve, the kind of physical compromise that altitude climbers recognize. But these 8 people were at 2,400 feet above sea level, in the Arizona desert, not on a mountain. By month 12, oxygen was at 17%. The decline was consistent, measurable, and accelerating. And here was the puzzle. If oxygen is being consumed, carbon dioxide is being produced. That is chemistry. You cannot burn through oxygen, metabolically or microbially, without releasing carbon dioxide. The equation is not optional, but the carbon dioxide wasn't there. The sensors tracked it. The atmospheric carbon dioxide levels were not rising in correspondence with the oxygen falling. Something was absorbing the carbon dioxide before it registered. A sink, invisible to the instruments, somewhere in the 3-acre enclosure. The team ran hypotheses: the ocean, the soil, some unusual metabolic pathway in the rainforest. None of the models could account for the scale of the discrepancy. By month 16, the oxygen was at 14.2%. That is the atmospheric equivalent of standing on the summit of a 4,000-meter mountain, Mont Blanc, the base of Everest's death zone. These 8 people were living at that altitude, 24 hours a day for months, while trying to farm, maintain complex machinery, and conduct research. The decision was made in January 1993. Oxygen would be injected from outside. The sealed world had to be opened. Jeff Severinghaus and Wallace Broecker were scientists at the Lamont Doherty Earth Observatory at Columbia University. They were brought in to solve the disappearing carbon dioxide problem, to find where the carbon was going. Their tool was isotopic analysis. Carbon dioxide is not one thing. The carbon atom at its center can have different numbers of neutrons: carbon-12, carbon-13, carbon-14. These isotopes occur in different proportions depending on whether the carbon dioxide came from biological processes, from geological sources, or from industrial combustion. By tracking the isotopic signature of the carbon dioxide inside Biosphere 2, Severinghaus and Broecker could trace where it was going. The analysis revealed 2 mechanisms operating simultaneously. The first was the soil. Before the sealing, Biosphere 2's soils had been enriched with unusually large quantities of organic matter, compost organic material, to create rapid fertility. The logic was sound: rich soil grows better crops. The unforeseen consequence: the soil's microbial population had an enormous food supply. Bacteria metabolize organic matter by consuming oxygen and releasing carbon dioxide. With an abundance of food, the microbes were working at many times the rate the models had assumed. They were consuming oxygen and producing carbon dioxide far faster than the plants could process it. This alone would have been serious. But the second mechanism explained the mystery. All that microbial carbon dioxide was not staying in the atmosphere. It was reacting with the concrete. Biosphere 2 contained vast quantities of concrete. In the foundations, the structural supports, the floors, the internal walls. Concrete contains calcium hydroxide. When calcium hydroxide is exposed to carbon dioxide, it reacts. Calcium hydroxide plus carbon dioxide produces calcium carbonate and water. This reaction is called carbonation. It happens in every concrete building in the world, slowly, over decades. In Biosphere 2, the carbon dioxide concentration was elevated, far above normal atmospheric levels because of the soil microbes, and higher carbon dioxide concentration drives the carbonation reaction faster. Severinghaus and Broecker calculated that carbonation inside Biosphere 2 was proceeding at approximately 10 times the rate it would in a normal building, exposed to ordinary air. The concrete was absorbing carbon dioxide and locking it into calcium carbonate, mineralizing it, hiding it in the walls. The total carbon dioxide absorbed by the concrete was estimated at approximately 750 kilomoles, a vast quantity, enough to account for the oxygen deficit. The oxygen had not disappeared. It had been converted, via microbial metabolism and concrete carbonation, into water and calcium carbonate, and deposited, molecule by molecule, into the walls, floors, and foundations of the structure. The plants could not replace the lost oxygen, because the carbon dioxide they needed for photosynthesis was being consumed by the building before it could reach them. No ecological model created for Biosphere 2 had included this variable. The concrete had been treated as inert, as structure, as background, as given, not as a reactive participant in the atmospheric chemistry of a closed system. This was not an engineering failure in the conventional sense. It was a gap in a model, an omission of a process that occurs in every concrete building on Earth, that had simply never mattered before, because no one had ever tried to run a closed ecosystem inside one. The oxygen injections, in January and August 1993, resolved the immediate crisis. The 8 biospherians completed their 2 years and exited on September 26, 1993, exactly on schedule. But the sealed world had not been sealed, and the oxygen problem was not the only thing that had gone wrong. 40% of the approximately 3,800 species introduced into Biosphere 2 went extinct during the first mission. Virtually all pollinating insects died. The bees collapsed early, within the first months. Without pollinators, crops had to be pollinated by hand. The team spent hours each week moving pollen between plants with small brushes, doing the work that a functioning ecosystem performs invisibly. The 4 species of cockroaches imported to decompose organic waste were overwhelmed. Domestic cockroaches had entered the structure as stowaways before the sealing, and exploded in numbers that threatened the crops. The team managed but could not eliminate them. The dominant ant species was not one of the cataloged ecosystem participants. It was the longhorn crazy ant, a globally invasive tramp species that had infiltrated before closure. It outcompeted every other ant species and established numerical dominance. Useful, unstoppable, not in the plan. The tropical rainforest had a different problem: morning glory. The vine grew at rates the models had not anticipated, climbing over and blocking the light to other plants. Without manual intervention, constant trimming, redirecting, management, the rainforest would have been lost to a single species of decorative vine. The desert biome had become too wet. Unexpected condensation patterns in the enclosed atmosphere were depositing moisture in what was designed to be an arid zone. The ecology that had been planted for dry conditions was adapting, imperfectly, to wet ones. Only 40 to 50% of the sunlight outside the structure was reaching the plants inside. The steel structure and glass panels filtered more light than the models had predicted. The plants were photosynthesizing in permanent partial shade. In each of these failures, the pattern was the same. The models had been good, the planning had been careful, the knowledge was genuine. But closed ecosystems at this scale behave in ways that open-air models cannot fully predict, because open systems leak. They absorb unexpected inputs, they export unexpected outputs. The variables that a closed system traps, carbon dioxide, species interactions, light levels, interact in ways that only become visible when there is nowhere for the consequences to go. Biosphere 2 was widely described as a failure. That is the incomplete account. What the experiment proved was specific and valuable. A sealed ecological system of 1.27 hectares cannot currently be made self-sustaining over a 2-year period, using the modeling tools and ecological knowledge available in 1991. That is not a failure of ambition. It is a result. It is precisely the kind of result that a Mars mission would need to know before departing. The concrete finding, that building materials participate chemically in the atmospheric system of a closed space, was not known before Biosphere 2 made it visible. Every closed habitat built for space exploration since 1993 has had to account for this. The finding has shaped the engineering of every subsequent sealed environment designed for human habitation. The microbial soil finding, that organic enrichment accelerates microbial metabolism at rates that overwhelm photosynthesis, has become foundational to how researchers think about soil management in closed systems. The ecological findings, that certain resilient generalist species will always dominate a closed system, that pollinator populations collapse rapidly in the absence of the environmental cues that regulate them outdoors, have informed decades of research into what it would actually take to sustain a diverse ecosystem in an enclosed space. Biosphere 2 has been owned by the University of Arizona since 2011. It operates today as an active climate research center. The original biomes remain: the tropical rainforest, the ocean, the savanna. Experiments are run on how soils respond to warming, how rainforests react to drought, how coral reacts to ocean acidification, at a scale and under conditions of control that no outdoor field site can offer. The name was Biosphere 2 because Biosphere 1 is Earth. The goal was to build a second one. What the experiment revealed instead was how much we don't understand about the first one. 8 people sealed the doors in September 1991, intending to demonstrate that we knew enough to build a world. 2 years later, they came out with a list of everything we hadn't known to ask about. In the practice of science, that is not a failure. That is a first result. The question is what we do with it.